All right, so tonight we're going to talk about chapter 27, and we're going to hit the highlights of this chapter. This is going to be the last lecture for the semester, and we're going to talk about acid-base and fluid balance. And so a lot of this will be reiterative of what we've seen before, and some of it will be relatively new. But one of the first topics I'd like to introduce are the fluid compartments of the body. So when you take a typical person of about 60% water by body weight, and that can vary anywhere from 50 to 60% water. A lot of it depends on body composition. So for example, someone with a relatively high muscle content would store more water. Fat, on the other hand, is hydrophobic, and so water is not stored in fat. So someone with a relatively high body fat composition would have a lower water by body weight. But for our purposes, we're going to take someone with 60% by weight water, and we're going to look at the distribution of water within the body. So we have got what we call the 60-40-20 rule. We have 60% of the body water by weight. When we look at where this water is distributed in the body, about 40% is within the intracellular fluid. So that is contained within the cells of the body. Now the intracellular fluid makes up one of our two major compartments of the body, our two major fluid compartments. We have the intracellular fluid and we have the extracellular fluid compartment. There are two main subdivisions of the extracellular fluid compartment. We have the interstitial fluid, and we have blood plasma. We also have some minor compartments of the body where we have fluid that is not really much in circulation with these, so they're sort of self-contained. So we have the cerebrospinal fluid, lymphatic fluid, the serous fluid within our pleura, our pericardium, and our peritoneal membranes. We also have synovial fluid, we have the perilymph and endolymph of the ear, and we have the aqueous humor of the eye. And this would be the minor fluid compartments. Minor fluid compartment, I'll just say FC, minor fluid compartments of the extracellular fluid. Our two major fluid compartments of the extracellular fluid that we'll be dealing with is the interstitial fluid and the blood plasma. So additionally, about 80% of our extracellular fluid will be contained within the blood plasma, and about 20% within the interstitial fluid. And what we're going to see is that we're going to have within the extracellular fluid, we have particular ion concentrations of different anions and cations, and that will be relatively uniform across our extracellular fluid compartments with some minor differences. And we're going to have in the intracellular fluid a different set of cations and anions. So let's take a look real quick. So when we look in our intracellular fluid, when we look at our major cations, we're going to see that we're going to have basically magnesium and potassium. So magnesium and potassium are some of our major cations or positively charged ions we see in cells. We also will see in cells that one of our major anions is phosphate. And we get phosphate, as we know, from molecules like ATP, adenosine triphosphate. So this is one of our major anions. And then we have some negatively charged proteins within the cell. And this is another one of our major anions. Now, when we look at the difference in composition between the ions within cells and outside of cells, we will see that the majority of our cations in the extracellular fluid are going to be things like sodium. And where sodium is, chloride follows, so chloride will be one of our major anions. We'll also see things like bicarbonate ion in here. Bicarbonate ion is another one of our anions. And we will also see that we have in the blood plasma, we're going to have some negatively charged albumins, again, proteins. And these are specific to the blood plasma. We do not find them in the interstitial fluids because, as you remember, the capillaries only allow small solutes and ions to get through, so the larger albumins stay behind, and they do not get into the interstitial fluid. And for this reason, we'll see a slight difference between sodium and chloride in the interstitial versus the blood plasma, because these negatively charged anions tend to drive chloride ion, into the interstitial fluid while drawing potassium into the blood plasma. So we have a little bit of variation, a little bit more sodium in the blood plasma, a little bit more chloride ion in the interstitium, but by and large, the compositions of the fluid between the interstitial fluid and the blood plasma does not vary that much. And these are both the major compartments of the extracellular fluid. The intracellular fluid, on the other hand, has a different set, as we've seen, magnesium and potassium, of cations and our anions, phosphate, and some of these negatively charged internal proteins. Another thing we always have to be aware of is that we've got our sodium-potassium pump always working. So it's always kicking out 2 sodium and bringing in 2 potassium. So 2 potassium and be kicking out 3 sodium. And as you recall, this is constitutively active, and it's driven by ATP because we're moving these ions against their concentration gradients. And this is one of the things that's helping to keep the composition of these different ions within their respective compartments. So let's now look at one of the other things that's very important in here is we're going to see that some of the way that the fluid composition in the body is maintained is by monitoring what's going on within the extracellular fluid. The body has a way of monitoring the composition of the solutes and the fluid volume of the extracellular fluid, not directly but indirectly, and can make adjustments. So if we get dehydrated, for example, and we lose extracellular fluid, especially from the blood plasma, then we can draw some fluid from the intracellular fluid into the extracellular fluid. But we have to have either a change in fluid volume on one of these compartments or the other, or we're going to have a change in solutes. Either way, if we keep all things the same, if our solute concentrations are the same, and we increase the plasma, then with the increase in volume of the plasma, that would cause a decrease in the concentration of our solutes. And so we have to remember that we measure these solutes in osmolality. So basically, number of constituents of solute that we have within a liter. So typically we measure them in milliosmoles, as you remember, milliosmoles per liter. And so we would look at the osmolarity, and what we'll find that within the extracellular fluid, blood plasma and so forth, it's almost always 300 milliosmoles per liter, between 290 and 300. Anything that gets outside of this range can start to be a danger zone. So what we're trying to do is maintain our concentrations at about 290 to 300 milliosmoles per liter. And as you remember, osmole, just to remind you, is the amount of solute particles per liter. So if we had sodium chloride and we put 1 mole of sodium chloride in 1 liter, we would get 2 osmoles because we would have 1 mole of sodium and 1 mole of chloride, right? And if we have, well, let's just use more physiologic concentrations. Let's say we had a millimole of, let's just say, calcium chloride. So we would have 1 calcium and 2 chlorides. And if we just threw this into a liter of water, we would now have 3 milliosmoles per liter, because this thing would dissociate into a calcium ion and 2 chloride ions. So this is just a quick recap of how we measure the solute concentrations in fluid, and we always have to remember that we're going to be wanting to keep these concentrations around this value, and anything that gets severely out of this value or starts to deviate out of this value can start to have severe consequences. So we will see, as the body is making adjustments, we can have fluid shifts from one compartment to the other. Now let's talk about some of the ways that ions and water move. Obviously, we have
selectively permeable membranes. So all of our plasma membranes are selectively permeable. Things can get in via diffusion, via this facilitated diffusion, or sometimes by active transport. One of the things that we have already seen is carrier molecules. For example, are sodium potassium ATPase, that in this case is an active pump. It's going to move things against their concentration gradients. We have transporters that can move things with their concentration gradients, and we've seen this before. And we've also seen how water can move by osmosis. And water will always move by osmosis, so it's not going to be actively transported anywhere. It's just going to go down its concentration gradient whenever we have a membrane that is permeable to it. And as we've seen, in some cases, we can adjust the permeability of certain membranes. For example, in the distal convoluted tubuli in the collecting duct, we can add aquaporins into the membrane to make them more permeable to water, and that way we can resorb more water out of the tubular fluid. Now, one of the things we talked about when we talked about the extracellular fluid is the fact that it's somewhere around 290 to 300 milliosmoles. And not surprisingly, because the osmolarities, because we have osmosis across these cell membranes from the cellular compartment to the extracellular compartment, not surprisingly, the solute concentrations within the cells are going to be the same. So they're going to be the same, and we're going to have an adjustment of fluid so that if there's a drastic change in one of the compartments, then we'll have a fluid shift. All right, so let's just talk about some of the major points here. So all the homeostatic mechanisms, as we've said already, that monitor and adjust the body fluid composition are going to be responding to the changes in the extracellular fluid, not the intercellular fluid. This is very important. As we said, we don't have receptors that directly monitor the fluid or electrolyte balance. However, they do have indirect means of monitoring this. We also know that cells don't move water by active transport. Everything is moved via osmosis. And finally, the body's electrolyte or water content will always rise if you have more input than output. So if you have dietary gains in water and you drink a lot of water, well, then you're going to have that much water in the fluid. And so typically, to maintain homeostasis within the duration of the day, you're going to excrete or get rid of as much fluid as you intake. And if for some reason you should dehydrate and not take in enough fluid more than you excrete, well, then this will be a way that we will dehydrate. And we'll talk a little bit about that in a minute. All right, so let's talk first about some of our hormonal regulations. We've talked about this before. This is nothing but recap. We have antidiuretic hormone. As we know, antidiuretic hormone is made in the hypothalamus and secreted by the posterior pituitary in response to a low blood volume. So this means we have low plasma. So typically our solute concentrations would be high relative to the volume of the water, the plasma, which is 95% water. It's going to be relatively high compared to the volume of water. So that means that we want to increase our plasma volume. So we would increase our antidiuretic hormone, and that would cause the insertion of those aquaporins in our distal convoluted tubules and our collecting ducts, so we resorb more water out of tubular fluid and return it to circulation rather than excreting it as urine. We have aldosterone. This is made within the adrenal cortex, and this is going to cause the kidney to conserve sodium ions. So as we know, where salt goes, water follows. So aldosterone is going to operate some pumps or I should say, put some pumps into the distal convoluted tubuli, the distal part of the distal convoluted tubuli, and the collecting duct. And that's going to have some sodium pumps that are going to pump sodium actively out of the tubular fluid back into the interstitial fluid, where it can be picked up again by the capillaries and returned to circulation. Now, opposing these two, we have our natriuretic peptides. And natriuretic, as the name implies, means excreting sodium. So this was when we have a blood volume that is high. And so this is going to cause there to be a stretch on the walls of the heart, so the aorta and the ventricles. And when they experience that stretch, which means we have a high blood volume, they will basically produce these natriuretic peptides that will cause the kidney to excrete the sodium and also to not to reabsorb water. So basically it will be peed out in the urine. All right, so as we've seen, the antidiuretic hormone causes water conservation at the kidneys. That's going to reduce our water loss. It also helps concentrate urine. So what else does it do? It's going to stimulate thirst. This is going to make us want to drink more. Obviously, if we need to replace the fluid, then we need to stimulate thirst centers to make us want to drink more water. All right, so antidiuretic hormone, you also have to remember, is also called vasopressin, not to forget that. And that can also, if it gets in really high concentrations, can decrease the luminal diameter of blood vessels and increase peripheral resistance. So we have osmoreceptors in the hypothalamus. These are monitoring the osmotic concentrations in the extracellular fluid. So if we have a change in the osmotic concentration, this is going to alter the osmoreceptor sensitivity. And then if we have particularly high concentrations, then we can secrete antidiuretic hormone. All right, so as we remember, we had those axons in the anterior hypothalamus that are going to release the ADH into the neurohypophysis, that's the posterior lobe of the pituitary gland. And there again, the rate of release is going to vary with the osmotic concentrations as measured by the hypothalamus. And that, of course, is going to be dependent on the concentrations within blood plasma. Aldosterone, as we remember, is secreted by the adrenal cortex. This can be secreted due to changes in potassium levels, increasing levels of potassium, or decreasing levels of sodium in the blood system, or in the blood plasma, I should say. It can also activate the renin-angiotensin-aldosterone system. So remember, aldosterone is part of the renin-angiotensin system. So what does renin do? Remember, it's released by the juxtaglomerular complex, and it's going to cause the conversion first of angiotensinogen into angiotensin 1, and then angiotensin 1 will be converted to angiotensin 2 by our ACE converting enzymes, as we've discussed before. Angiotensin 2 will have some of its effect on the vasculature system, but it will also have some effect right there on those arterioles coming into and leaving the glomerulus, as we have talked about before. Now, we also will determine the rate of sodium absorption and potassium loss along the distal convoluted tubuli. Oftentimes, the absorption or resorption of sodium is at the expense of potassium. So you're either going to gain sodium back and spit potassium into the tubular fluid, or vice versa. And this will occur along the distal convoluted tubuli in the collecting system, as we've seen. As we know, water follows salt, so particularly water follows sodium. So if we have the kidney conserving salt, then water will follow. So if salt leaves, or I should say if sodium leaves the tubular fluid and goes into the interstitium, then water will follow. And both of those will then end up back in circulation. All right, natriuretic peptides have the opposite effect. As we said, these are released by the myocardium in the atria and the ventricles. So these will reduce thirst because we don't want any more plasma volume than we've already got. It will also block the release of antidiuretic hormone and aldosterone, and it causes diuresis. And what is diuresis? Diuresis is basically losing water through urination. So if you've ever heard of the term diuretics, if you want to get rid of excess water in your body, people take these things called diuretics, which supposedly causes you to urinate more often and get rid of more water. But basically the natriuretic peptides, that is their job. One of their jobs is to cause diuresis, to lower the blood plasma levels, or I should say the volume of plasma. All right, now we can look at the movements of water and electrolytes. So when the plasma volume decreases and the electrolyte concentrations will rise by virtue of the fact that we have the same number of electrolytes we started with, now in a smaller volume. And when the body loses electrolytes but doesn't lose water, then the electrolyte concentrations will decrease because we put them now in a larger volume of water. But typically when the body loses electrolytes, water will follow and is lost by osmosis in most of the systems. So we will have different regulatory mechanisms that are going to monitor the losses of water versus the loss of electrolytes. All right, so fluid balance is going to be basically maintained between the extracellular fluid and the intracellular fluid. And remember that we want to keep our osmolarity the same within both compartments. So basically fluid can shift from one compartment to the other. As we know, water circulates freely in the extracellular fluid compartments. So basically, remember, that's the blood plasma in the interstitial spaces. So we know at capillary beds that we have the hydrostatic pressure forcing water out into the plasma out of the plasma into the interstitial spaces. The water will be resorbed along the distal portion of the capillary bed, where our oncotic pressure, which is our colloid osmotic pressure, is going to pull some of that water back into circulation, and the rest ends up going through the interstitial fluid, returning to the lymphatic circulation, which will eventually be dumped back into venous circulation. So as we say, the extracellular and intracellular fluids are in osmotic equilibrium. And when things start to differ between them, then we have to have some kind of mechanism to return them to homeostasis. So we should not have any super large scale circulation between the components. All right, so when we look at the movement within the extracellular fluid, here we're talking about blood plasma and the interstitial fluid. Basically, we've talked about this a million times before. You can go back to our capillary diagram. We've got the hydrostatic pressure pushing the water out of the plasma and into the interstitial fluid. And we have the net colloid osmotic pressure from those albumins that are in plasma, pulling some of that water out of the interstitial fluid back into plasma. Now, as you recall, we lose more fluid into the interstitium than we pull back into the plasma, but eventually that fluid does get returned into venous circulation via the lymphatic system. All right, so when we have the extracellular fluid volume being redistributed, that is, as we've said, once again, from the lymphatic system to the venous system, so back to the plasma. And so we basically have this continuous fluid filtration, as we know. So the extracellular fluid volume is 80% in the interstitial fluid in the minor fluid compartments. I kind of flip-flop that back around in the previous part of the lecture. And 20% of it's in the plasma. All right, so if you get, for some reason, you get a collection of too much of fluid out of the plasma and into the interstitium, then we call that edema. And if for some reason this happens within the lymphatic system instead, this is called lymphedema. And this is basically when you have the blockage of lymphatic vessel or lymphatic drainage. And so some of that fluid is forced out of the lymphatic system into the interstitium. And so you can have lymphedema caused by that. All right, so when we think about daily fluid losses and gains, typically we lose about 2,500 milliliters of water each day through urination, through insensible perspiration, through vapor on our breath, feces, and so forth. We can have other problems cause water loss, like fever, also diarrhea, these kinds of things. We have sensible perspiration, that's sweat, and that can vary with activity. And of course, if we are sweating a lot, we typically take in a lot of water. So if we sweat a great deal and we don't take in water, we can start to become dehydrated. Typically, when we start to dehydrate, then the thirst centers are stimulated and we will start to drink more water. All right, so typically we will gain about 2,500 milliliters a day through eating, so that's about 1,000 mils or a liter a day. Through drinking, maybe about 1,200 mils a day. And also metabolic generation. This is happening within the cells themselves. And if you recall, the mitochondria are hard at work. And what do? Well, they convert basically glucose and fatty acids into ATP. And the byproduct of that is carbon dioxide and water. So water is one of the byproducts in about 300 milliliters a day through mitochondrial activity. So here's just some of the different ways that we can lose and gain water. So we've got some metabolic activity from the mitochondria through oxidative phosphorylation, producing about 300 milliliters of water, and that will go into the extracellular fluid. We can absorb about 2,200 milliliters across the digestive epithelium that will end up in circulation. We'll pee out about 1,200 milliliters of that. Some of that water will be secreted by sweat glands, some lost in feces, and some as water vapor, as we've talked about before, skin, lungs, and just breathing away the water. All right, so we talked about the metabolic generation of water. I'm not going to go too much into these slides, but this will bring us back into our fluid shifts. So if we have a change in the osmotic gradient between the extracellular and intracellular fluids, then we will have a rapid movement of water between them to stabilize or to equalize those osmotic concentrations once again. So if the extracellular fluid osmotic concentration increases, that means we have more solute to water, then the fluid is going to become hypertonic to the intracellular fluid. So what's that going to happen? That's going to cause water to move from the intercellular fluid into the extracellular fluid compartments. Now, if the opposite happens and we have the osmotic concentration increase, or I should say decrease now in the extracellular fluid, so if we get more water now and the solute concentrations, or I should say the solute numbers haven't changed, the concentration will now decrease because we had the same number of solute particles, but now we have more water. So now the fluid will become hypotonic to the intracellular fluid, and that's going to cause water to move from the extracellular fluid into the cells. So this would happen if you drank like 2 gallons of water. So this would be the scenario if you drank 2 gallons of water without sweating them off or getting rid of them. And this scenario here would happen if you started to dehydrate. All right, so typically, as we know, the intercellular fluid is a lot more than the extracellular fluid because, as we recall, 40% of our, from going back to our 60-40-20 rule, 40% of that body weight water is going to be retained within the cells, and the other 20% is in the extracellular volume. So our intracellular fluid is going to be this massive water reserve. And that's going to prevent a large osmotic change in the extracellular fluid, which, if it happened very suddenly, could kill us. All right, so what happens when we lose a lot of water? Well, we have dehydration, that's water depletion, and this is when we get water loss greater than water gain. So this is when you go out and run your marathon and you didn't quite drink enough. So your water's lost, but your electrolytes are retained. So basically your extracellular fluid osmotic concentrations are going to rise because you're reducing the volume, but you still have quite a few electrolytes in there. And so now the water is going to move from the intracellular fluid to the extracellular fluid. But because you haven't really moved a huge proportion, because remember you got 40% in the intracellular fluid, you don't have a huge change in the overall concentration. So the net change is going to be relatively small. So you could have severe water loss from excessive perspiration or just not drinking enough water. Vomiting and diarrhea are big problems when people are sick because when you vomit, you throw up a lot of water, and when you have diarrhea, you lose a lot of water through feces, especially if they're really watery and if they're really not solid. We could also have some physiological mechanisms to restore this balance. For example, antidiuretic hormone and renin secretion. Obviously, we're going to have to increase that volume as much as we can. So we're going to retain salt. Where we retain salt, water follows. We're also going to stimulate our thirst. And renin is going to decrease our luminal diameter of our peripheral capillaries, and that's going to raise blood pressure. And once again, behavioral changes, stimulating thirst to increase our fluid intake. All right, so now we can talk about what happens if we drink a bunch of water, but we don't drink any electrolytes with it. And then our extracellular fluid volume is going to increase. Now our fluid volume is going to be hypotonic to the intracellular fluid. Now fluid is going to shift into the cells. And if we have too much of that, we can have hyperhydration. And this is what's going to happen if you have, remember what happens if you put a cell in hypotonic solution, it will swell. If it goes too long, it can explode. So we've got our distortion of cells. And so this is going to cause problems because you're going to change the solute concentrations around the enzymes and disrupt the normal cell functions. Plus, eventually, you could actually have lysis of the cell. All right, so if you have too much fresh water with, let's say you drink distilled water and there's no electrolyte concentration in it at all. If you put hypotonic solution into the bloodstream, you inject it, there can be endocrine disorders, for example, excessive antidiuretic hormone production. There is an actual physiologic problem where you have too much antidiuretic hormone production, and this can cause overhydration. Or if for some reason you cannot eliminate excess water in urine due to renal failure or heart failure, so you're not pumping blood. Sometimes cirrhosis of the liver can cause this. All right, so a lot of times in overhydration, we're going to see a low concentration of sodium. This is called hyponatremia. And this is one of the things that we have, what we call water intoxication. And there were a few people who died of hyponatremia where they were going to run a marathon and they thought they needed to drink so much water, and they drank so much water that they actually had hyponatremia and they had abnormally low sodium concentrations because they had too much volume of water in the extracellular compartment. All right, so here is just a diagram that shows the shift from compartments. So here we have our intracellular fluid. Notice the volume is about 40% to 20%. Together they make up 60% of our total water volume in the body. So our intracellular fluid are balanced here. Our concentrations of ions are going to be the same per unit volume. Now, let's just say we lose some water from the extracellular compartment. Maybe we sweated it out or something, but we didn't sweat out a bunch of electrolytes with it. So now what's going to happen is we have a hypertonic solution over here, and water is going to follow solute. So water is going to shift out of the intracellular compartment into the extracellular compartment, and we'll have a little bit of a decrease in the intracellular fluid compartment and a little bit of an increase in the extracellular fluid compartment. And notice we have lower volume of water altogether, in the body, but now the volumes, the osmolarities are balanced between the two fluid compartments. So we now have the same solute concentration on this side as we do on this side. All right, so when we keep our electrolytes in balance, we have to gain and lose electrolytes in the same amounts. There again, it's a matter of keeping homeostasis. So whenever we get some, then we have to get rid of some. So basically, we have a constant turnover of electrolytes, and that's going to affect, obviously, the water balance. And the concentrations of individual electrolytes can also affect cell functions. So as we know, sodium is dominant in the extracellular fluid. And so it's going to give 90% of the extracellular fluid osmotic concentration. So sodium chloride and sodium bicarbonate. So basically, when we have our bicarbonate ion here, we can have a sodium ion on it. And this would be baking soda pretty much. So now that we've covered the fluid compartments of the body and the fluid shifts in the osmolarities and concentrations within the fluid compartments, let's move on or shift our focus to the acid-base balance. So obviously we have to be able to control our pH and maintain it within the homeostatic range. So typically that's between 7.35 and 7.45 within the ECF, and we usually measure this at the plasma. It's the easiest to measure. And anything outside of this becomes dangerous. So anything below 7.35 becomes acidosis, and anything above 7.45 is moving towards alkalosis. And of course, we're going to have acids and bases that are dissolved in body fluids, as well as salts. And these things are all going to affect the pH. So acidosis is basically a physiological state resulting from abnormally low plasma pH. Again, basically below 7.35. Alkalosis is above 7.45. So we've already gone over that. Acidosis and alkalosis can be of two forms. We can have a respiratory acidosis or alkalosis from either not clearing enough carbon dioxide from the breath, in which case we would get acidosis from a buildup of carbonic acid, or if we exhale too much carbon dioxide and bring in enough oxygen, we could start to get alkalosis. But the more dangerous type of acidosis and alkalosis are metabolic acidosis and alkalosis. All right, so basically both of these will affect all body systems. We're going to see nervous and cardiovascular systems affected first, and of course these, since they're central to regulating other systems, if these systems start being affected, then all the other systems will start to fall along with them. And so both are dangerous. Acidosis is more common, but in either case they can be quite dangerous, and being able to determine whether somebody has acidosis or alkalosis and whether it's respiratory or metabolic are key factors in treating someone who's coming into the ER or into treatment with some kind of metabolic or respiratory problem. All right, let's talk about the types of acids in the body. We have fixed acids, organic acids, and volatile acids. Fixed acids are ones that, once they're produced, don't leave the solution. They will remain in the fluids until they are eliminated by the kidneys. Some examples are sulfuric and phosphoric acid, and these are ones that are generated during the metabolism, typically the catabolism, or breakdown of amino acids, phospholipids, and nucleic acids. Organic acids are produced by aerobic metabolism, so think lactic acid. And these are pretty rapidly metabolized and not accumulated. So when you think about when you make lactic acid, what happens to it? When you make it at the muscle, it's shuffled away, picked up by circulation to the liver as part of the Cori cycle, where it's then assembled into pyruvate, then glucose, shipped back to the muscle so that it can then make glycogen. So organic acids are very rapidly metabolized. And finally, we have a volatile acid, and that would be carbonic acid. And as you know, carbonic acid is formed when carbon dioxide and water come together to form carbonic acid, which then dissociates into bicarbonate ion and hydrogen ion. And so we see that a very powerful enzyme in this conversion is called carbonic anhydrase. And this can catalyze this reaction really in either direction, depending on where it's located. So we will see that we have carbonic anhydrase in red blood cells, as you recall, where it converts the carbon dioxide and water into carbonic acid, so it can then be dissociated into bicarbonate ion, and the hemoglobin buffer can pick up the hydrogen ion, and that's how carbon dioxide is carried around in circulation typically. And we also saw it in the kidney cells. We saw it being very important for regulation of the bicarbonate ion in the kidney. We see it in parietal cells in the stomach and all over the body, really. So basically, the carbon dioxide and the amount of carbonic acid is going to be related to the amount of carbon dioxide in circulation. So basically, the partial pressure of carbon dioxide. So the partial pressure of carbon dioxide is going to affect the pH in body tissues. So as carbon dioxide rises, the partial pressure of carbon dioxide rises. That means things are going to become more acidic, and that means the pH is going to go down. So the partial pressure of carbon dioxide and the pH are inversely proportional. So at the end of the day, basically, we've got to have the number of hydrogen ions and the number of bicarbonate ions balanced. So basically, we're looking at making our gains and losses of hydrogen ions balanced and our gains and losses of bicarbonate ions balanced. So hydrogen ions, as we know, we can gain them by eating them or through cellular metabolic activities. We can eliminate them at the kidneys and we can eliminate them at the lungs. But they must be neutralized in order to avoid tissue damage. If we get outside of our normal physiological range, then bad things happen. Protein starts to denature, enzymes stop working, and so forth. So we have some safety methods. We got some buffer systems. And buffer systems, we've talked about them before. If you remember that very first lab we had at the beginning of the semester, we talked about 3 specific buffer systems. And these are basically weak acids, and they're what we call conjugate bases. So it's a way we can shuffle hydrogen ions back and forth. So we can either absorb them when we need to take them out of solution, or we can release them back into solution if we're getting too alkaline. So buffer systems, as we know, is going to consist of a weak acid and the anion released by its dissociation, i.e. carbonic acid. You have hydrogen ion released and the bicarbonate anion. And so the anion is what we call that weak or conjugate base. So in the solution, molecules of weak acid are going to be existing in equilibrium with their dissociation products. Exactly what we said earlier about the carbonic acid bicarbonate ion. Basically, that reaction going in opposite directions at the same rate, so that we have a balance of these ions in equilibrium. All right, we've got three major buffer systems that we're going to talk about here: the protein buffer system, the carbonic acid bicarbonate buffer system, and the phosphate buffer system. And since we've already been over them in some detail a little bit earlier in the semester, all I'm going to do is highlight where we're going to see them most active. So, for example, we can see the phosphate buffer system is going to be very important in the intracellular fluid. So think about where we find phosphate in the intracellular fluid. So think about our ATP and other molecules that are going to be having phosphate as a component of it. So phosphate, as we saw earlier on, is one of our anions in the intracellular fluid. Now, the protein buffer system is going to be in operation both in the intracellular and the extracellular fluid. And we've got three different variants of the protein buffer system. We've got the hemoglobin buffer system, which is in the red blood cells. And this we saw was very important as we bring carbonic acid into the red blood cell, because it will dissociate into bicarbonate ion, and then our hydrogen ion will then be buffered by the hemoglobin buffer. So it will be picked up and sort of neutralized by the hemoglobin buffer. Then we've got our plasma proteins. That's going to be our albumins and plasma proteins and such. And they, like all proteins, are going to have a carboxyl end, a carboxyl terminus, which can act as a carboxylic acid and donate a hydrogen ion. And they're going to have an amino end, which can act as a base and sometimes pick up a hydrogen ion. And amino acids themselves also have a carboxyl end and an amino end. So these are what we call amphoteric. And we've seen this before. We've seen how amino acids can be both acids on their carboxyl end. The carboxyl group can donate a hydrogen ion or release a hydrogen ion and become a carboxylate ion. And then the amino end can pick up that hydrogen ion. And in physiological pH, that's typically what we see happen. So it becomes an amphoteric molecule, in that it can act both as an acid, as a base, and it's also what we call a Zwitter ion. So it's hybrid. It can be negative on one end and positive on the other. So the protein buffer system is going to be comprised of these three components, and we find it active both in the intracellular and extracellular fluid. Now, finally, the carbonic acid bicarbonate buffer system is going to be at play in the extracellular fluid. So this is where we're going to see these three buffer systems and where we're going to see them operate. Now, some limitations on the carbonic acid bicarbonate buffer system is that what it does is it's going to prevent pH changes caused by organic and fixed acids in the extracellular fluid. One of its limitations is it cannot protect against rapid changes in CO2. So the carbonic bicarbonate acid buffer system cannot protect against CO2, since CO2 is one of its major components. So that is one of the things about the bicarbonate buffer system. And one of the rate limiting factors is how much bicarbonate do we actually have. So bicarbonate availability is part of the sort of rate limiting factors of the carbonic acid bicarbonate buffer system, and it also only functions when the respiratory system and their respiratory control centers are functioning normally. Now, When we think about bicarbonate ion, we rarely have a shortage of it because when we have an abundance of it, it will usually form sodium bicarbonate. So we'll have the bicarbonate ion pick up a sodium ion, and now it's basically baking soda. So we have this bicarbonate reserve so that, for example, if we are low on bicarbonate ion, we can just simply pick it up from our sodium bicarbonate reserve over here. So here we can see the whole process, and it's usually in equilibrium. This part is in equilibrium. So we have the carbonic acid, carbon dioxide, carbonic acid, bicarbonate is going to be shuffling these hydrogen ions back and forth between carbonic acid and bicarbonate. And remember that we have bicarbonate being one of the major ways via which carbon dioxide is carried in circulation. So here we can see bicarbonate ion picking up hydrogen, dissociating back into water and carbon dioxide, and then carbon dioxide can be breathed away at the lungs, and the excess hydrogen is now bound up in water. So that's basically the overview of how these buffer systems work. And since we've been over them before, and this is mostly review, I'm going to stop here. And from here on out, we'll be review and getting ready for the last lecture test and the lab practical.